Okay, let's get started, please. All right, so today's topic, can I have your attention, please? Thank you. Today's topic will be receptor tyrosine kinases, a different type of receptor than what I covered last time. But before I get to that, there's a few more slides at the end of the last lecture, so we're going to go over those, and then we'll head into today's main topic, which will be a shorter lecture, I think, based on the amount of stuff that's in there. Okay, so this is where I left off last time. As you all were hurriedly trying to get out of here, I was trying to convey my excitement about new ways to stop asthma and how modifying a binding or encouraging the binding of a ligand to a receptor can really help with therapies. So that's just to remind you where we left off. So what I want to do to finish off the last lecture is to sort of provide more context for these GPCRs we talked about, G-protein coupled receptors. We've been using adrenaline as one of the examples of how these ligands and receptors work. And I wanted to put this slide up because this is something you learned about in the first half of class, exocytosis and endocytosis. And when you learn about these things at first, it's just like, well, here's this process, memorize it, here's what it does, right? It's really nice to be able to integrate how this is happening with what you're learning now. None of this is happening in a vacuum, it's all related. So I want to show you how that works in this example. If this is a cell that's secreting adrenaline, what actually happens is that the cell builds up a supply of adrenaline inside of it in vesicles so that it's ready to release those very quickly, because as you know, the flight or fight response is very fast. And so these vesicles basically hang out near the cell membrane, and when they're stimulated, they release the adrenaline. And that happens, as I mentioned last time, in your adrenal glands located here, right near your kidneys. So these vesicles are in chromaffin cells in the adrenal glands. But we don't want adrenaline being released all the time. That would be quite something if we were always amped up like that, right? So how do you regulate this process? You don't want these vesicles coming out at any time. Turns out that some of the receptors we've learned about are involved in this process. Ion channel coupled receptors play an important role in regulating how adrenaline is actually released. And so if you look at this, I've taken this diagram now again and just filled in one more of the generic spots with something specific. So let's put the calcium channel in here. A calcium channel is an ion channel coupled receptor that in response to a signal that we're not getting into today is going to open up and cause calcium to come inside of the cell. And then calcium in turn regulates the release of these vesicles. How does that happen? You've actually learned about that previously as well. It happens through the fusion of these vesicles with the membrane of the cell in this process that you learned about where these proteins are winding together like ropes, and they come together and they turn around tightly, and the vesicle pushes up against the membrane, overcomes the activation energy, fuses, and releases its contents. So what's calcium doing in all of this? Well, synaptotagmin, which I think you learned about, I hope you did, is one of the proteins involved in this process, is a calcium binding protein. So here's a vesicle approaching the plasma membrane. This would be the outside of the cell down here. And as that vesicle gets close, and this snare-dependent vesicle fusion uses all these proteins to get everything to come together and fuse, synaptotagmin, one of the core proteins responsible for this, will only do it once it's bound to calcium, shown by these red dots here. So then calcium-dependent vesicle fusion is how adrenaline gets released. So I want to now kind of put all that together for you, again, as a concept to understand pathways, not as something I expect you to go memorize, but you should be able to understand these types of integrations of pathways and how they work together. So let's start from the beginning then. Of course, there's always a beginning before the beginning. I'm not telling you what opens the calcium channel, but let's start at the calcium channel opening. Here in your adrenal glands, you have cells. Those cells, when necessary, open up a calcium channel, an ion channel coupled receptor, to let calcium in, which then binds to a protein, changing its shape as we've discussed, causing it then to stimulate the snare complex mediated fusion of vesicles with the membrane, which in turn then releases the ligand adrenaline outside of the cell, which then through endocrine signaling, as it's a hormone, goes throughout your body, binds to G protein coupled receptors that respond to adrenaline, that then starts the cascade you learned about last time, where those GPCRs are activating G proteins, adenylyl cyclase, cyclic AMP, protein kinase A, and one of the important end results of this process here, which I told you about here, is bronchodilation. opening up your respiratory tract, your respiratory tract so that you can, excuse me, breathe better. So that kind of takes you through an example of how you can go from a signal using one type of receptor, causing changes in protein shapes, leading to the expulsion of another ligand, which then binds to GPCR and causes a really important process inside of you to happen. And all of this, of course, is happening on a very fast time scale. So something to think about, and maybe something that helps you kind of put all these different things together. So far then, in this first part of the class, we've talked about ligands and receptors. We've talked briefly about ion channel coupled receptors and about GPCRs in a fair amount of detail. This is the main pathway I've focused you on. Here it is, including its inhibition with a phosphodiesterase, which is able to kind of inhibit this pathway by converting cyclic AMP into AMP. So all that information is here as a starting point for you to review, and of course the details are in the slides I taught you about. Shaded out here is what we haven't discussed yet. Today we're going to cover in detail receptor tyrosine kinases, in particular 2 of the pathways that they work through. And then before we move on, the last slide of this previous lecture, the slide that many of you have been waiting for, I'm sure, is what's the summary of what you have to learn so far? Here it is. So I'm not going to read each of these to you because it's redundant, but there it is. It's in your lecture capture. It's in your PDF. If you are wondering what should I be studying, go to this slide. It really summarizes the main things we've covered and what you do need to know and what you don't really need to know as well. So for example, this is the pathway that I've told you several times I haven't really covered in class, but you should be generally aware of it because you'll see it on your MCATs, your DATs, your GREs, et cetera, et cetera. On the other hand, if I tell you you need to know this pathway, you should know this pathway, right? There's no surprises here. Okay, so with that, we are going to move on to today's topic, receptor tyrosine kinases. Any questions about GPCRs or ion channel coupled receptors before I move on? Okay. No, okay. All right, so again, my recap is perhaps a little bit redundant since I just gave you slightly a recap, but previously on the HIT show, Cell Biology, we covered ion channel coupled receptors, GPCRs, and actually I'm going to play this movie for you again right now because we have time in today's lecture, and this concept is so complicated, I think another review doesn't hurt, so I'll go ahead and play that for you again right now.
Many G-protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate. Activating ligands remain bound.
Okay.
Many G protein.
Well, that's good. And then I already covered this today, so I'm going to skip that. And then we just talked about this. So now we're going to move on to enzyme-coupled receptors. Some difference between terminology I want to make clear up front. You'll see these terms quite a bit, enzyme-coupled receptors, and then the other term, receptor tyrosine kinases, or RTKs. To avoid any confusion, I want to make it clear up front. Enzyme-coupled receptors are a pretty broad class. It's what it sounds like. When you're coupled to something, you're kind of working with it or stuck to it. So an enzyme-coupled receptor is working with an enzyme. It's a pretty vague description, right? Receptor tyrosine kinases are a type of enzyme-coupled receptors, and it's the only type we'll talk about in this class. It's the most common type, and that's what we're going to focus on today. So I'll be saying RTKs or receptor tyrosine kinases a lot, but I think the vast majority, if not all of what I talk about, applies pretty generally to enzyme-coupled receptors in general. Now, I haven't taught you anything yet about RTKs, but if I tell you it's a receptor tyrosine kinase, and it's an enzyme-coupled receptor, if you take that information together, you can probably guess what it does. So does anybody want to take those names and tell me what you think it's doing? It's an enzyme-coupled receptor, and the name is receptor tyrosine kinase. Take a guess. Broadly speaking, yeah.
So this is the receptor that contains kinase which adds phosphate group to the activity?
That's exactly right. You had something as well? Yeah, so both of these are correct. So it is, you'll see what you're sort of on the right track actually, because it brings pieces of the receptor together, and then you mentioned it's kinase that's going to phosphorylate proteins. And the tyrosine part of the name tells you that it phosphorylates on the tyrosines. So this tells you what it does. It's a receptor that has kinase activity associated with it that's going to phosphorylate proteins on their tyrosines. So you already know quite a bit about what these receptors do, and I haven't even told you about them yet. The name often gives you a lot of information. So let's talk about how this works, and this gets to your comment about bringing two pieces together. Enzyme-coupled receptors in general, the way they work is that you have a signal here, and the signal in this example is a dimer, so it's got two pieces, light red, dark red. And here in the cell membrane, you've got two pieces of the receptor that are hanging out. They're inactive. They're not doing anything. When this ligand binds, it actually helps bring these pieces together. And when they're brought together, that bringing them together and the shape changes associated helps to activate the receptor. So now the catalytic domain, remember catalysis is encouraging a reaction to move forward. A catalytic domain encourages a reaction. This catalytic domain here is now active and it's going to do something. So that's the very fundamental aspect of how these enzyme-coupled receptors work. And they're called enzyme-coupled because of what I'm going to tell you next. So you could kind of view this as coupling and activating this enzyme, this domain here that's going to have enzymatic activity. Or the other way this can work is a signal brings these two pieces together, but then the two pieces that are the receptor have an enzyme nearby that binds to the receptor now and is activated. So in this case, it really is enzyme coupled in the sense that here's the receptor, here's the enzyme, and they're coupled. All right? So these are two different ways. In other words, the receptor can have its own activity as an enzyme, or it can bind to a nearby protein and activate that enzyme. Either way, it's an enzyme-coupled receptor that has activity. Now, you may already notice this is not that different from how GPCRs work. The mechanics are different, right? There's not two pieces of a GPCR that are brought together. That doesn't happen. But the end result where you have a ligand binding to a receptor and then something downstream is activated and turned on is the exact same thing here. So the broad concepts are the same. The details are different. And if you think about that as you move forward through this lecture and compare it to GPCRs, the compare and contrast might be a good way to learn, asking yourself, okay, what's different here? What's the same here? It'll help sort of sear the information into your brains about how these different types of receptors work. All right, so receptor tyrosine kinases have 20 subfamilies, so there's a lot of different types, and I expect you to memorize all of them. Kidding. Okay, so, but what I want you to take away from here is just how diverse they are. These different colored pieces represent domains of the proteins. You'll notice that all of them have this red domain, or two red domains, inside of the cell. So here's the cell membrane. That red domain is the tyrosine kinase domain, hence the name receptor tyrosine kinase. the kinase that phosphorylates at tyrosines. That's the consistency amongst all these different types of RTKs. And then on this side, the side that's exposed outside the cell, you've got this variety of domains that do different things. And that then speaks to the diversity of signaling that that RTKs do. We'll learn about some of these different receptors throughout the rest of the class, not all of them, but a few of them. For example, VEGF is the VEGF receptor is one that really helps stimulate blood vessel growth. Something we'll talk about when we get to cancer. And there's many other examples. FGF receptors stimulate different types of growth. And there's actually a trend here that a lot of these receptors, if not almost all of them, I think, are responsible in some way for stimulating growth or activity. And you'll see that as we go through today's lecture. So activated RTKs then, once you're turned on, right, this is the part we already talked about, ligand binds to receptor, turns on the catalytic activity, which is a kinase activity because it's a receptor tyrosine kinase. Once that happens, what happens next? Well, these RTKs recruit a complex of intracellular signaling proteins. And again, if this is all I told you, you could replace this with GPCR and be the same thing because, of course, these receptors are signaling through intracellular signaling proteins. But again, the difference here is in the details. So once you have these activated kinase domains, what do? Well, quite a lot of the time they phosphorylate themselves at those tyrosine residues. So if you zoomed in, you would see that, as happens, proteins have a bunch of tyrosine amino acids, and wherever this domain can reach, it'll put this phosphate groups, it'll phosphorylate these tyrosines on itself and on the nearby piece as well. So the whole receptor starts to get self-phosphorylated. Now that, as we've talked about, causes a shape change, and that shape change allows things to bind to it. So now it becomes kind of sticky, if you will. These are the light blue, whatever color that is, this dark blue. These are examples, generic examples of intracellular signaling proteins. These proteins hang out inside of the cytosol at high concentrations, not really doing much of anything in this context until you have this phosphorylation. Now the binding site is available, and they'll bind to the receptor. Once they bind to the receptor, their shape changes, and their activity, shown by these dashed lines here, is turned on. So now they have some sort of activity, and they can continue the signaling cascade down, right? So again, not that different from GPCRs in the sense that something is binding to something and changing its shape and activating it and so on and so forth, but the details are a little bit different. So each phosphorylated tyrosine then, as shown here, serves as a specific binding site for a different intracellular signaling protein. There's a type of domain called an SH2 domain that you can think of as like a docking site. So these proteins that bind here, they all have SH2 domains, and those domains are the domains that will bind onto the receptor where it's phosphorylated. And now I'm going to give you a lot of this information in a movie because I like to do that. That way you have me talking and then a movie as well. But yes, question?
Does it mean that all these intracellular signaling molecules that bind to phosphate receptors are positively charged?
Does that mean that all of these proteins that are bound to the receptors are positively charged? That is a fantastic question, and the short answer is I don't know. I would suspect not. I think it's probably a diversity of proteins. But I'd have to look that up. I can look it up later and let you know. Yeah, it's an interesting question. Yeah, I don't know. Okay. Yes.
Are they all going to be the same on either side? Like, one correlation.
Yeah, that's a good question. Are they going to be the same on either side? So here, you've got the same protein here and here, here and here and here and here. The short answer is depends. In this example here, because you're seeing what are supposed to be two identical pieces, then that implies that the binding sites are the same and the structure is the same, and so you would assume that the same proteins would bind because it's the same type of it's the exact same piece basically, but on two sides. Now there's a couple of caveats to that in real life. One is that you can have these pieces coming together that are not exactly the same, and that might then encourage different types of proteins to bind to one side or the other. And then the second caveat is that in real life you may have more than one protein that can bind to a given site. And in fact, that competition of proteins, so imagine if you had this light blue one, but you also had a green one or something that could bind to the same site. Perhaps that competition between them binding, or which ones are being made in a given cell versus a different cell, actually changes the net output. And is another way for the cell to control the output from the signaling pathway and customize it to what it needs. So in theory, yes, but in reality, there's probably a lot of complexity to that. Yeah. Any other questions? All right, let's watch the movie, which is...
Receptors, shown in orange, bind to the extracellular domains of their receptors. The receptor monomers dimerize. The cytoplasmic tyrosine kinase domains of the receptor monomers are activated, and tyrosine residues of the cytoplasmic domain are phosphorylated. The phosphotyrosine residues lie within amino acid sequence motifs, which are recognized by SH2 domains of several cytoplasmic proteins, shown in green. The receptor tyrosine kinase may phosphorylate the bound proteins, creating additional docking sites for SH2 domain-containing proteins, which themselves regulate the activities of other proteins. In this way, a signal cascade is initiated.
Okay. So the next thing we'll talk about are a couple of examples of this. So one example, the first one, involves the monomeric GTPase RAS. So I haven't told you about RAS yet, but you already know what a GTPase is. What's the most prominent GTPase that we talked about last week? Yes, G-alpha, that's right. Remember, G-alpha, when you have GPCR-based signaling, is a GTPase, because once it's activated with a GTP bound to it, it pretty quickly turns itself off by having that GTPase activity and turning that GTP back into a GDP. So you already learned about GTPases, but you learned about them with GPCRs, and now we're going to talk about them with RTK. So you're seeing a trend here. Cells, our biology, nature loves to reuse ideas and concepts over and over. There's a lot of redundancy and a lot of reuse of some very basic concepts throughout. So you'll see these things again and again. All right. So here's what's happening. You have your activated receptor here with the signal bound to it. You have these adapter proteins that we just talked about that are binding to the phosphorylated sites and being turned on. And then this is the downstream cascade, or the first part of it, involving the GTPase RAS. Now, before I take you through it, point of confusion that often comes up in this nomenclature, and I can't change the names of these things, unfortunately, but we have this RAS protein in green, and in blue we have a different protein called RAS-GEF. They're not the same thing. One is RAS, the other is RAS-GEF. And you may recall you also learned what a GEF is, a guanine exchange factor, which we talked about again with respect to GPCR-based signaling. But what a guanine exchange factor does is the pseudo-opposite of what a GTPase is doing. A guanine exchange factor is promoting the switching out of GTP and GDP. So take the GDP out and put the GTP in. Do you remember in GPCR signaling, what was the guanine exchange factor that was promoting this activity? Pardon me? It does. It exchanges guanine. So what was the exchanges these GTP and GDP. But what was the protein or molecule with GPCRs and all the things we covered? That was the GEF, the GEF there. Pardon me? The GEF. What was the GEF? So let's look back at that pathway real quickly. This is a good time to review. All right. So here's that GPCR pathway. And we're talking about taking the GDP out and putting a GTP in. So let's start with where that happens here. Where is that happening? The G-alpha. So what's promoting that change for the G-alpha? In other words, what causes the G-alpha to do that? The activated receptor. There you go. So this is actually, if you look at the slides, it's listed in there that the receptor is a GEF. So, right, so these are the concepts to start thinking about. In this pathway, the receptor acted as a GEF to switch this out in the G-alpha, turning the G-alpha on, and then the G-alpha was a GDPase. In this concept today, entirely different, we have some of the same general ideas, but in a different context. In this context, the RAS-GEF, this protein, is the GEF. In other words, the receptor is not the GEF, the way it was with GPCRs. And in this case, you do have a GTPase, and it's RAS. It's not G-alpha, it's RAS. Now, it may sound like I'm making things a little bit more complicated right now because I'm going back and forth. Oh, look what this did, look what that did. If you think about this and you go back and you review them, it will help you. If you start to think about the concepts and then applying them and plugging them into these different pathways. Long term, it'll make it easier to understand the material. So give it a shot. All right. So with that, we'll focus just now on this RTK pathway involving RAS. So you have this RAS protein. It's hanging out at the membrane. It's got this wiggly line here indicating that it's stuck in the plasma membrane. And once you have this receptor activated, this RTK, it is promoting the RAS-GAF. It's activating the RAS-GAF, and it's promoting this exchange of GDP coming out and GTP going in. And just like you learned about before, molecular switches, remember, GTP goes in, it usually turns things on. The RAS protein is now on. All right? So the RAS protein on is now going to have its own activity. What does it do? We'll talk about that in the next slide. So here's your activated RAS protein. And that protein is going to activate a series of comically named, but at least easy to remember, enzymes. MAP kinase, 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 MAP kinase, kinase, and MAP kinase. All right, it's not very creative, but it's pretty straightforward. So, if you've got a MAP kinase, the kinase that phosphorylates a MAP kinase is called MAP kinase kinase. That's basically the idea here. So through the signaling cascade, you've got a series of activations that get carried down. This is your cascade of your signaling pathway, your intracellular signaling proteins, ending up at the end here, vaguely defined, in a series of changes in activity. So these would be sort of your effector proteins that we've learned about. And look how they're divided up, fast changes, slow changes. It's those concepts from before coming back. So you have fast changes where you take existing proteins and you're modifying them, and you can have slower changes where you actually change the expression of genes in the RNA and the protein. All right. Oh, the name MAP kinase, mitogen activated protein kinase. Thinking ahead to upcoming lectures, based on what you've learned about the term mito, what do you think this might be encouraging? Okay, I heard some good answers in there. So mitogen, mitosis, the idea of promoting cells to grow and divide. This kinase helps promote cell growth and sometimes cell division. So again, there are often clues in the names of these proteins about what they actually do. And we'll come back to that later. Okay. So try to keep our phones off, please. Thanks. So the end result here is a normal signaling cascade, right? But things can go wrong sometimes. RAS, as it turns out, is often an issue in a wide variety of cancers, which we're going to cover in detail towards the end of class. But for now, I want to introduce to you what happens when you have RAS on too much. So one of the things that can happen is you might have a mutation in RAS where it's not able to hydrolyze its GTP. In other words, its GTP-ACE activity is not there anymore. And if you can't turn this back into GDP, then you're always on because you have a constantly bound GTP. And if you're always on, you're continuously transmitting information downstream. And if your role is to help encourage cells to divide or grow, you can start to probably appreciate how this could be a problem for putting you at risk of cancer because now your cells are on hyperactive drive and just dividing or growing all the time. So yeah, I guess I already said this, but mutations where RAS is always on are associated with cancer, uncontrolled proliferation, even in the absence of mitogens. Remember a mitogen here? A mitogen is a ligand that's going to cause or promote cell proliferation, which we will cover in a few slides down the road. Okay, so that's the monomeric GTPase RAS-based pathway for activation. The second pathway I'll tell you about today involves PI3 kinase and lipid docking sites. So the first part of this slide is exactly what you've been seeing. Here's the receptor, it's activated, it's phosphorylated, things are going to happen. Now instead of what I showed you here, instead of RAS-GEF being involved, this pathway involves PI3 kinase, hence the name, PI3 kinase mediated pathway. So now you have this PI3 kinase, and again, the name tells you it's a kinase, it's going to phosphorylate something. so that once that PI3 kinase binds to this receptor, it's activated, and that activation leads it to phosphorylate something called inositol phospholipid, which is a membrane-bound protein hanging out here, and that phospholipid, when it's phosphorylated, is now going to be able to bind to various protein kinases that in turn activate an AKT kinase-mediated pathway. Yes? Sorry? Oh, yes, absolutely. Yeah. So for simplicity, these diagrams are just shown in this direction. But yeah, you can pretty much imagine the other side being available to do the exact same thing, again, with the same caveats we talked about before. Yeah. So, oh, and I forgot to bring the candy today, so I'll bring it next time and then, grab it. I forgot to bring it, sorry. Okay, so AKT. AKT is a very commonly used kinase that, again, phosphorylates a bunch of stuff, and we'll talk about that. So what happens is AKT is hanging out in your cytosol unactivated until it is activated by these kinases, which are being activated themselves because they're binding to this phosphorylated inositol phospholipid. So again, activation of the receptor, activated PI3 kinase, phosphorylated inositol phospholipid, other kinases are activated, and they activate AKT. What does AKT do? Once AKT is activated, notice that all this stuff here is at the membrane. Hence, we have this in the title, lipid docking sites. So what happens is sometimes you have these sites where you can get proteins to come together and concentrate so that they're able to start the signaling cascade. But of course, whatever you want to happen inside of your cell is inside the cell. Either it's in the cytosol or it's in the nucleus if you want to change transcription. So to get from there downstream is where these signaling molecules you keep learning about come in. And in this case, AKT is one of the major components that helps relay that signal, because it's not bound to the membrane. It binds to this membrane-bound protein, and then once activated, goes and carries the signal forward. What does it do? AKT is most commonly involved in helping cells survive. So what you're looking at here is activated AKT encouraging a pathway. Remember, when you have a protein on top of an arrow, it means it's encouraging that arrow to go in this direction. All right, so it's catalyzing this reaction in a sense. And so what it's doing is it's encouraging this to happen instead of this. So let's talk about what these things are. BAD is a very appropriately named protein, BCL2 associated DEF promoter. All right, so DEF is bad. Right? You can kind of remember it that way. BAD would promote death, which is generally considered bad. So BAD is inactive, or rather it's bound to an inactive protein called BCL2. Once this survival signal comes in, it separates these proteins and activates. Now BCL2 can be activated. It changes its shape, it becomes active, and BAD falls away, it's inactivated. So AKT is phosphorylating BAD and causing it to fall away. Activated BCL2 protein promotes cell survival by inhibiting apoptosis. All right? So let's look at what happens then step by step. When you have this complex here, you're not promoting cell survival. You're encouraging apoptosis. The default is the cell dies. Remember I showed you that slide at the beginning that the default situation for cells is if you don't get the right signals, you will die. It's a safety mechanism. This is a very specific example of that. The role of these proteins is to not encourage cell survival until they're told to do so by a signal. In the absence of a signal, the cell dies. If the signal comes through and inactivates this guy, and this is able to now be active, it's going to promote the cell survival and stop the cell from dying. And another way to look at this, by the way, is this is an example of that whole inhibitor of an inhibitor concept I told you about very vaguely before, where you have, let's see if I can draw, where you have you have basically cell survival. Well, you have apoptosis being inhibited. So here's apop. This is really bad. All right, my kid can write better than that. Okay, so you've got apoptosis being inhibited by until you have this inhibition of BCL2 being removed. I'm not going to try to write this out, but let me just, you know what? Okay, so basically you have inhibition of BCL2, and once you have this removal of it, now this is active and it's able to promote cell survival by inhibiting apoptosis. So you have a couple of different parts here of inhibition and activation, and if you try to put it together, it'll kind of make sense. I hope I didn't just confuse everybody by trying to draw that. Any questions about this or anything I should clarify? Because I don't know if I just made things worse. Yes? Yeah, that's a great question. Can it be too late to survive? So what if the signal binds to it too late and the cell doesn't survive? Right, so the way you can think about this is that these types of signals for cell survival are pretty constant. And it's really the removal of the signal that causes the problem. So in this case, in this pathway example, we've described the pathway as, oh, look, AKT is coming in and then promoting cell survival. But in real life, if you will, the AKT signaling promoting cell survival is going to be on most of the time. Right? It's kind of the default. So like the on state is kind of the default, and then when you turn it off or don't have it, then the cell might die. And this goes back to one of the earlier things I had mentioned where these on and off states are equally important, or, on isn't always the most important. It really depends on what they're regulating. So yeah. Yes? So the question, can I describe at which step of the inhibition this promotion occurs? I won't, because it's actually a lecture that's coming up in Unit 4 that's all about apoptosis and all these pathways. So just for the sake of having to divide things up, I have to leave you a little bit hanging on this. But we will talk in detail about apoptosis and what happens. So you're going to see these proteins again in Unit 4, BAD and BCL2. Yeah. So stay tuned. Yeah. Anything else? Yes?
Since apoptosis and cell survival are equally important, which state is it contingent on the BCL2?
So yeah, that's an interesting question. So, since cell proliferation and apoptosis are equally important, broadly speaking, in what state do you find BCL2? And the answer is it really depends. You're going to find it in a variety of states in different cells. Apoptosis, as we'll learn about later, is actually a very normal part of what happens to us every day. So every day we're making millions and millions of cells, and we're killing off millions and millions of cells. If I scratch my skin, I just had a bunch of cells that fell off. Side note, which I love to gross people out with, dust in your home is mostly parts of you and your pets, cellular debris that's fallen off of you. That's what dust is mostly composed of. So think about that next time you're cleaning up. But getting back to your question, then apoptosis is very much a normal, important thing. So in a given cell, whether it needs to be living to do its job or being killed off sometimes to do its job or because there's something wrong with it, that state of BCL2 will change quite a bit. The other part of the answer to your question, which is kind of outside the scope of this class, but just to properly answer it, is that in reality, these things, I show you a single protein and here's what it does. Of course, that's not how it's working. Inside of a cell, you actually have a constant tug of war going on between levels of different things. So you might actually have a bunch of BCL2 molecules, and some of them are active and some are inactive, but maybe the active ones are being bound and sequestered away, or they're not in the right place to do the next thing. And maybe the inactive ones are quickly coming up in volume and they overtake. Maybe something else, it's really dynamic. And what actually happens is the end result is a result of which side of that tug of war wins. So it's really more complicated, so that's why it's an interesting question. Yes? Yes, the death rate for different cells is very different. Neurons versus, let's say, skin cells or something else. The way to think about it is that these pathways are the end effectors of a really important decision, and different cells will have different times and places and priorities for doing that. So obviously, your neurons, you want to preserve them for the most part. On the other hand, you may or may not know this, but we'll cover it at some point, probably in apoptosis. When you're born, you make a lot more neurons than you need, and then you prune them out to actually make the pathways that connect. Yeah. So a child who's growing up is actually undergoing selective apoptosis to make the neural networks make sense. So it's pretty, yeah. Yes?
For cancer growth, is it absence of BCL2 or is it inactivation?
So for cancer growth, is it absence of BCL2 or inactivation? Again, it depends. A lot of the issues that we have with cancer come from multiple mutations that can overactivate a protein or inhibit a protein. Depends on the context. So if you have a protein here that when active promotes cell survival, then its role in cancer, if it's mutated, would probably be to increase its activity. But again, we'll cover that in detail in the cancer part of class. Yes?
So are signals constantly coming in, or like is this like a selective switch is kind of staying on?
So are signals constantly coming in, or is the switch staying on? Is that the question? Yeah. In this example here, to survive, the signal has to constantly come in. So if you were to get rid of it, well, it's a little bit of both. I mean, so let's imagine the signal stops, right? What's going to happen is that you have this inactivated BAT already and this active BCL2, so they'll stay active for a while because they're already done. But no protein hangs around forever, right? And for all we know, and I'm not getting into it, there are probably regulatory mechanisms for getting rid of this protein quickly and making more of it so that you keep it constant and fresh in a sense and sort of keep the signaling where you want it to be. So as this state of it goes away, this signal will become more important because if you're not making more of this phosphorylation, then eventually you'll have a problem. So it's a little bit of both. Like it's not going to be an immediate difference, but the signal has to really be there to maintain the signaling state. Yeah. These are great questions, guys. Keep them up. All right. Anything else before I move on? Okay. All right. So experimental science can inform what we learn. I mean, obviously, everything you're learning about was discovered in a lab somewhere at some point. That's how we know these things. But these examples here from your book are really useful for thinking about signaling pathways and mutations in those pathways, et cetera, et cetera. So let's, it's a little simple exercise. Let's assume that you have here on this side a signaling pathway involving 3 proteins, RAS, which you learned about, and then proteins X and Y. When you don't have any signaling, nothing's happening, right? Now, let's say you have the cell that has a mutant protein X, and it's somewhere in there. You don't know anything about X yet. And when you don't have any signaling through RAS, no problem. But when you introduce overactive RAS, that's always on, signaling is restored. Now, for the sake of this exercise, assume X and Y are in the same pathway as RAS. So if X is in the same pathway as RAS, and it's mutated so that it's not doing what it's supposed to do, that's I guess the caveat there. It's not doing what it's supposed to do. And then you introduce overactive RAS, and signaling is restored. Is X upstream or downstream of RAS? Who thinks it's upstream? Raise your hand. And who thinks it's downstream? Raise your hand. A lot of people didn't even raise their hand. Let's try this again. Come on, guys, wake up. If it's upstream of RAS, raise your hand. And if you think it's downstream of RAS, raise your hand. Okay, anyone want to offer any? Well, so I'll tell you the answer. The answer is that X is upstream of RAS, and here's why. Imagine X and RAS as being part of that river of flow, right, the signaling pathway. Imagine where you're putting the dam in place in the river to block the water flow. So if you have a signaling pathway that has a mutant protein X, in other words, that's where the dam is going into the system, and that protein is upstream of RAS, if I block the water upstream of RAS, but then I start pouring water into the system downstream where RAS is, the signaling can move forward, right? That makes sense? So let me try to draw this out. Yeah, I know. I'll try to do a better job drawing. So here's your water flow going down this river, all right? And let's say RAS is over here with an R and a line apparently. So let's say the answer, remember the answer is that X is upstream of RAS. So let's put X up here. So here's your water flowing. Oh, RAS is mutated. RAS is mutated. X is mutated. So X is mutated and the water stops right here. No more water flow goes and the river dries up. But now if I add overactive RAS here, in other words, I dump a bunch of water into here, water flow is restored. Okay? So you know that X is upstream of RAS because putting in a bunch of RAS fixed the problem. It's downstream in the pathway that's carried the signal forward. On the other hand, with mutant protein Y, no signaling because Y is not working. You introduce overactive RAS and still no signaling. So Y is upstream or downstream of RAS? Downstream, right. So Y is somewhere down here. And what's happening here is that even though you're adding an overactive RAS, you're dumping all this water in here, it's not going to do anything because Y down here is going to stop the water flow. And so the signaling cascade, again, hits that dead end. So think about pathways as flows of information or water and what's happening when you have a mutation somewhere in the pathway and the effect it has. And that helps you figure out. This is actually, by the way, how people figured out these pathways, by doing these experiments and understanding what's upstream of this or downstream of that based on what happens. Okay. How do I get, okay. All right. Erase all that. All right, there we go. So this tells you the order of the proteins. So now with this information about X and Y, you know that this receptor is working through RAS, and that X must be something here, and Y must be something here. X is upstream, and Y is downstream. And then, if you really wanted to, based on what you know about the RAS pathway, you could probably start taking guesses, if you looked at that previous slide, as to what X and Y actually are. So that's kind of gives you an idea of how this all comes together. All right, what's the end result? Usually, RTKs are responsible for promoting cell growth, and sometimes cell proliferation. Depends. So a growth factor in this example is binding to this receptor. It's taking us through this cascade that we just covered. A note about nomenclature. Notice how there's like multiple arrows here. That's done a lot when people draw pathways to indicate that there's multiple steps here that are not indicated in the diagram. Just again, something to be aware of. People will give you signaling pathways throughout your life, and that's what that means. So basically, what this is telling you is, long story short, you're activating something called TOR, a protein we haven't covered, that then leads through a variety of processes to increased cell growth. So going back then to this slide of the GPCRs and the RTKs, I've now taken you through a number of these pathways, right? We covered this pathway in detail. I kind of mentioned this one to you, but didn't cover it. And then today we've covered this RAS-based pathway and then this PI3 kinase-based pathway. These are kind of major things to review and to get on top of and to understand. All right, the next thing we're going to talk about are receptors that go straight to the nucleus. This is a unique class. And the example I'm going to give you, the most common example that people talk about, and it's in your textbook, is delta notch signaling. Delta notch signaling works differently. It's not a GPCR, it's not an RTK, it's not an ion channel receptor. What it is you have a cell expressing this delta ligand in red, and the ligand is membrane bound. And then you have a delta receptor in green here, also membrane bound. They have to make contact. So remember the one of the first slides about endocrine signaling, paracrine signaling, neuronal synaptic signaling, and the fourth one was contact-mediated signaling. This is contact-mediated signaling. The ligand is bound to this membrane, and the receptor is bound here, and they have to touch. Once they bind, the interesting thing is that the receptor itself chops part of itself off and sends it straight into the nucleus to activate transcription. So this is sort of like the shortcut way of signaling, right? There's no downstream signaling proteins, no intracellular signaling cascade, nothing. There's not even an adapter protein that's binding and being activated by this receptor. The receptor is activated, cuts itself in half, not technically half, but cuts itself. This piece goes in and turns on transcription. Really simple, effective, and fast signaling pathway. Hey folks, I can hear you when you're whispering and talking, especially if it's loud, and that's going to be distracting to your colleagues as well, so please keep it down. Thank you. Okay, the next topic I want to briefly tell you about is cell-cell communication being different in plants and animals. The vast majority of this class, in fact, probably everything except for the next few slides, is about what animals do, what vertebrates do. That tends to be mostly of most interest to students and also of most relevance for your next steps in your career. But it's good to know what other types of species do as well. So plant and animal cells have been separated in evolution for more than 1 billion years. That's a lot of time to start doing things differently. And they've solved the issue of being multicellular in distinct ways. And I want to pause and explain that comment. Everything we've been talking about in some way or the other, when we talk about cell signaling, why does it matter? I mean, if you're a single-cell bacterium, you will sense your environment, but you're not communicating with other cells. But if you think about everything we've talked about today, it tends to be in a multicellular context. You have to be able to talk to each other if you're one of trillions of cells to really make the system work, right? So how do you solve that? Well, we've talked about GPCRs and RTKs and notch delta signaling and ion channel coupled receptors. These are ways to solve that problem. Plants also use transmembrane receptors, mostly enzyme coupled, but there are actually few or no RTKs, GPCRs, or steroid type of receptors. So they have receptors that are mostly enzyme coupled. In other words, they're very similar to RTKs, but they're not RTKs. And this is why it's important. Remember I mentioned to you at the very beginning, enzyme-coupled receptors is a big class, and RTKs is the most common example, blah, blah, blah. Well, this is why I said that. There are examples, like in plants, where you have enzyme-coupled receptors that are not RTKs. And I wanted to share this with you from Wikipedia because it's kind of cool. There's a plant hormone ethylene that's been used since the ancient Egyptians, who would gash figs in order to stimulate the fig's ripening. Wounding the fig would stimulate ethylene production by the plant tissues, which would then activate receptors that would promote ripening of the fruit. The ancient Chinese would burn incense in closed rooms to enhance the ripening of pears, because the incense, as it turns out, they didn't know this, but the incense has the same ligand that would do that job, so it has ethylene in it. In 1864, it was discovered that gas leaks from street lights, like in the streets of London, etc., led to stunting of growth, twisting of plants, and abnormal thickening of stems. And it turned out again that those gas leaks had ethylene, and that ethylene was binding to these receptors and causing changes in the plants. So kind of neat to think about. How does all of this work? So ethylene is the example we're going to use. If you don't have any ethylene, here's your cell membrane receptor. Again, it's an enzyme-coupled receptor in plants. And in the absence, and this is where it gets a little bit funny compared to what you've learned, this receptor is active without a signal, right? Not with a signal. So in the absence of any signal from ethylene, this receptor is on, as these lines indicate here, and it is activating a protein kinase, which is in turn causing a transcriptional regulator to degrade away, and so the genes here are off. So ethylene-responsive genes are off, even though the receptor is on. And you could draw this out again with the arrows and the upside-down T's. I'm not going to try again. But basically, if you draw this out, you're talking about activation, but the end result is off because this is, you could think of this as an inhibition, because this regulator that's needed here is being degraded. Now, when you have ethylene present, it binds to the receptor, and it turns the receptor, quote-unquote, off. And this is, again, why it's so important not to just think of things as on and off. Each of the states can be really important. In this case, this off state of the receptor actually ends up turning the transcription of the genes on because of the way it works. The receptor is off, the kinase is off. Because the kinase is off, it does not phosphorylate this regulator, and because the regulator is not phosphorylated, it's not degraded, it survives, it can go to the nucleus and cause transcription. So if you looked at this big picture, you'd say, okay, no ethylene responsive genes, ethylene responsive genes. But the mechanism is different, and it's just a difference in how the plants evolved their use of these receptors. All right. This next point I'm going to share with you, I never took our Twitter break. Let's do that right now. Science Twitter. No, discard, because I think I can see some sleepy folks. All right. Since we have some time today, I'm going to share two with you. One is a receptor tyrosine kinase. So since we're talking about those today, the video isn't like the most amazing, but it's kind of neat to see. So let me explain to you what you're going to see. This is from a paper where they looked at dissociated hippocampal neuron culture. So they took neurons out from a mouse, put them into a dish, and then put into those a couple of things they wanted to watch. They put in something called KIF1B beta, EGFP. You're going to learn about this family of proteins later. Don't worry today. This is for fun. But basically, long story short, this is a protein that's going to carry things along a component of the cell, along I'm trying not to tell you too much too early, but basically it's involved in cell transport. It's going to carry things along this element of the cytoskeleton, okay? And that's in green. And then what they did was they used a red fluorescent protein, RFP, to tag a receptor tyrosine kinase. And that's going to be in red here. And then they have the merge below that. So what they're showing, if you watch this, and they're going to point to an example here, this transport protein moving along the cytoskeletal element here, it turns out is carrying one of these receptor tyrosine kinases to a different location. So something we haven't even touched upon, receptors being moved to different parts of the cell. It's just kind of a fun thing to think about, how complicated things really are versus the basics I teach you about here. And then a cooler video that has nothing to do with today's lecture, but that I'm super excited about. Jan Huisken, who's at University of Wisconsin, and does some wonderful imaging of zebrafish embryos, and I work with zebrafish myself. He's He'll actually probably visit here in a few months to give a talk, and if anybody's interested, I'll let you know about that. But anyway, he designs really neat microscopes. And what I'm going to show you is a time-lapse video of a zebrafish embryo over many hours compressed in time, where the zebrafish nervous system is going to develop. It's monocolor, so you're going to see a single color here, and you're going to see a bunch of what look like neurons coming out, and they actually are neurons. The head of the fish, well, let me make sure, orient you guys, yeah. So the head of the fish is going to be over there, and then this is the spinal cord, and the tail is going to be back there. And because this is so cool, I'm going to make it full screen and start it from the beginning. Yes, okay. All right, there you go. So these are axons and dendrites coming out from neurons, and this is the spinal cord forming. These are other cells moving around. The yolk would be over here. A lot of these are probably immune system cells moving around. And he's going to turn it so you can see a little bit better from this side. That's the neural network forming in a vertebrate embryo. And now you can see the developing spinal cord pretty well. These little circles here are individual cells. All right, so that's kind of cool. I hope that woke up everyone a little bit. That's a live imaging of a zebrafish embryo developing and its nervous system developing, so kind of fun. All right. All right, back to the not as exciting lecture notes. Well, we're almost there. All right, so the next thing I want to tell you about is a very generic statement, but it's something your book emphasizes, and we should at least kind of cover it in some detail. Protein kinase networks integrate information to control complex cell behaviors. You actually have already learned this. I've kind of been telling you this, but now it's formalized in this very super simplified slide here. You have ligands and receptors, and then you've got all these cascades we're talking about. And the only point of this slide is that things are much more complicated than what I present to you so far. A given pathway with PI3 kinase or a given pathway with RAS, these things tend to converge and integrate downstream as well. And in fact, that review slide I have of ion channel coupled receptors and GPCRs and RTKs, if you look at it, you'll even see it there. You'll see arrows going from side to side a little bit. So just remember for yourself that these kinase-based networks, they are complicated, they're integrated, and that we try to give you the basics of the understanding, but of course in your cells there's a lot going on as these things talk to each other at multiple levels. And this is an example of that. Again, don't try to memorize this, but this is just to kind of give you a sense of inside of a cell, and this is only a subset of all the pathways that are present, how you have changes in gene expression, or that can lead to changes in cell proliferation, the dividing of cells, cell death, and how dependent these things are on so many different types of pathways. Now here's what's kind of neat, for a basic cell biology class. This was, I think, our third lecture for this segment of the class, but yet you've already learned a fair amount of this. You've learned how to read signaling pathways. You have heard about RAS. You've heard about RTKs and the growth factors that can stimulate them. You've heard about hormones. You can guess what kind of receptor this is. What kind of receptor is this probably for a hormone? Huh? Or a GPCR maybe? Yeah, so it's probably a GPCR because hormones usually bind to a receptor at the cell surface. Survival factors, this is probably also a GPCR. Ion channels can also play an important role, absolutely. And so you're starting to learn about all these different components and how they come together. There's BCL2, for example. And as we go through the rest of the class, we're not going to learn all these things, but these types of diagrams will really start to come together for you. So the next thing I want to do, one of the last things I'll do today, is I often do try to bring it back to clinical relevance, as that tends to make it interesting, and maybe a lot of you have experience with something like breast cancer. One of the most common cancers, something that we all will know somebody who has it, whether it's a friend, a family member, or unfortunately yourself at some point, perhaps. And so let's talk about some of the therapies that have been really useful for that. If you're wondering why should I care about receptor tyrosine kinases, this is one reason why. So let's discuss what's written over here. HER2, also known as ERB2, is a human epidermal growth factor receptor 2. This is a receptor tyrosine kinase, and it would be present here on the outside of this cell. HER2 is a cell membrane surface-bound RTK, and it's normally involved in transduction pathways leading to cell growth and differentiation. That's probably not a surprise. You just learned about cell growth being dependent on RTKs. HER2 is upregulated by estrogen receptors. So another nearby estrogen receptor can upregulate HER2. And then HER2 then dependent breast growth is a normal part of development during puberty and pregnancy. So it has a very important normal role to play in women. Unfortunately, approximately 15 to 20% of breast cancers have increased expression of HER2. And based on what this says, as the states, increased expression of HER2 can lead to abnormal or faster cell proliferation. So again, something that's doing its normal job correctly in cancer can be upregulated or mutated or otherwise altered to do something it shouldn't do, and encourage, in this example here, too much cell proliferation, which can lead to a tumor. All right? So here's the good news. Since this is a significant portion of breast cancer cells, treatment for HER2 has been an interest of a lot of groups and companies, and there's been significant progress made with it. So what happens is that you have this cancer cell that's got too much HER2 expressed. Remember I told you about agonists and antagonists and inhibition, right? So what if you could find a way to block the signal going through HER2? If there's too much HER2 and you block some of that signal, perhaps you can then make it harder for these cancer cells to divide. With that concept in mind, a long time ago, a company came out with something called Herceptin, named after HER2. And the way it works is that Herceptin is, as shown here, is actually a HER2 antibody. It binds, as shown here, to HER2 receptors, blocking the receptor sites, and most likely therefore preventing further growth by interrupting the growth signal. And so HER2 can slow the progression of breast cancer disease. Right? So HER2 is acting here, as what type of a ligand? Is it an agonist or an antagonist? Antagonist. It's playing defense, right? The agonist for HER2 is trying to dunk the basketball, and all HER2 is doing is blocking it. You're not going to do that. So it's acting as an antagonist. And so an antagonist for receptor tyrosine kinase has helped a lot of people recover or lead better lives when dealing with a pretty serious disease. So, food for thought about the relevance of this class. Yes? What's the difference between an antagonist and a competitive inhibitor? So, an antagonist, the analogy. Well, let's do a numbers analogy today. If you have an agonist, it takes a signal from 0 to, let's say, 10. An antagonist would just block it, and the signal would stay at 0. An inhibitor, if depending on what you mean by inhibitor, if let's say it's causing the downstream effect to be inhibited, then you can think of the signal as going from 0 to a negative value. So like maybe it's actually reducing even the downstream signaling beyond what would happen in the absence of the ligand binding to the receptor. Yeah. I mean, the full answer to your question is a little bit more complicated. It's very context dependent, but that's kind of the idea. Any other questions? All right. So the last thing I'll leave you with today is another fun thing to share. We've talked in the last 3 lectures about how cells are signaling to each other. And it's all been about ligands and receptors. Turns out there's far more to our biology than that. And this is one example of it. The title of this article, if you'd like to go read it, it's in your PDF in detail. I'm not going to read the whole thing to you, obviously. It's an article about how the Internet of Cells has biologists buzzing. Networks of nanotubes may allow cells to share everything from infections and cancer to dementia-linked proteins. Now, this is a controversial concept and is being actively researched. It's this idea that people have seen this mostly in cell culture, that you will have cells that seem to create connections to each other, as shown here. And what do those connections do? I mean, you could argue that it could be a connection just like if I reach out and I tap somebody on the shoulder, maybe it's just saying, hey, hello. On the other hand, what this is actually suggesting, or a lot of groups are suggesting now, is that these are hollow tubes that can connect them and connect signals. So instead of sending a ligand out into the matrix to go, extracellular matrix, not the matrix from the Neo movies, into the extracellular matrix to go and bind to a cell, instead, what if you actually have a tunnel, and it's almost like a subway tunnel, and you just send your signal through that tunnel to get to the other cell? A direct cell-to-cell communication mechanism. There is some evidence for this, and then there's some evidence against it, so it remains controversial, but something to think about. And they have a nice YouTube video here that I'll play for you. It's only a minute long. right, so I'll leave you with that thought that as you think about everything you've learned about cell signaling, remember that there's so much more we still are figuring out, and maybe there's more to the story than we realize. All right, with that, I'll see you in a couple days.